Good day to everyone. We are group number 5 and we are going to report on applications of nucleic acid in biochemistry. The content of this report is about introduction of gene DNA cloning, genes to genomes, genomics and proteomics field, genetic engineering, and genetically modified organisms.
Introduction of gene. A gene is the basic physical and functional unit of heredity. Genes are segments of DNA that contains the code for a specific protein that functions in one or more type cell in the body. The human genome identifies the genes that contains about 20,000 to 25,000 genes of human. Genes carry the information that determines our traits, which features are characteristics that are inherited from our parents. Some traits are caused by mutated genes that are inherited or the result of a new gene mutation.
Good day everyone, I am Shelley and will discuss about gene or DNA cloning. By definition, the term cloning means obtaining multiple copies of something. To clone a gene is to make many copies of it. Gene or DNA cloning is a molecular biology technique that makes many identical copies of a piece of DNA, such as a gene. In a cloning experiment, a target gene is inserted into a circular piece of DNA called plasmid. A plasmid is a small, circular, and double-stranded DNA molecule and naturally exists in bacterial cells. Gene or DNA cloning can be achieved by two different approaches. Cell-based or in vivo, which involves the use of restriction enzymes and ligases using vectors and cloning the fragments into the host cells. And in vitro, or using polymerase chain reaction or the PCR method to create copies of fragments of the DNA without the need for host cells. Cloning vectors, DNA molecules used to transport clone sequences between biological hosts and test tube. Cloning vectors are plasmids that are used primarily to propagate the DNA. DNA cloning is the first stage of most of the genetic engineering experiments. Steps in DNA cloning. The DNA is cut from the source organism using restriction enzymes. Restriction enzymes, also called restriction endonuclease, is a protein produced by bacteria that cleaves DNA at specific sites along the molecule. The piece of DNA is pasted into vector. The ends of the DNA are joined with vector DNA by ligation. Ligation means to physically join a DNA vector to a gene of interest. The vector is now introduced into the host cell of either the bacterium or yeast by a process called transformation. The host cells copy the vector DNA along with their own DNA, creating multiple copies of inserted DNA. And the vector DNA is separated from the host cell's DNA and now purified. The DNA that has been cut and pasted from organism into a vector is called recombinant DNA. Other types of gene cloning. Reproductive cloning is the process of making genetically identical copy of an organism. Dolly is a female sheep and the first mammal clone, and it is the most well-known example. Therapeutic cloning is the process of making multiple copies of a cell to treat a disease. It is also known as the somatic cell nuclear transfer. Applications of gene cloning. Gene cloning is useful in studying the structure and functions of genes in detail. Medical applications. The clone bacteria plays an important role for the synthesis of vitamins, hormones, and antibiotics. And lastly, agricultural application wherein cloning in bacteria facilitates nitrogen fixation in plants.
Genes to genomes. Genes are made of DNA, and so is the genome itself. A gene consists of enough DNA to code for one protein, and a genome is simply the sum total of an organism's DNA. It includes both the genes, the coding regions, and the non-coding DNA, as well as mitochondrial DNA and chloroplast DNA. The DNA is long and skinny that is capable of contorting like a circus performer when it winds into chromosomes. The study of genome is called genomics.
Good morning, everyone, and good morning, Sir Terwin. So, my report for today is all about genomes. But before that, uh, the key experiments done between the 1920s and the 1950s established convincingly that this genetic information was carried by DNA. In 1953, with the elucidation of the structure of DNA, it was possible to begin investigating how this information is passed on and how it is used. Let's talk about genome. We use the word genome to describe all of the genetic material of the cell. The total amount of genetic information in the chromosomes of an organism, including its genes and DNA sequences. The genome of eukaryotes is made out of a single haploid set of chromosomes that is contained in the nucleus of every cell and exists in two copies in the chromosomes of all cells except reproductive cells. The human genome is made up of about 30,000 genes. Proteins make up body structures like organs and tissues, as well as control chemical reactions and carry signals between cells. If a cell's DNA is mutated, an abnormal protein may be produced, which can disrupt the body's usual processes and lead to a disease such as cancer. The primary function of a genome is to store, propagate, and express the genetic information that gives rise to a cell's architectural and functional machinery. However, the genome is also a major structural component of the cell. Besides its genetic role, the genome affects cellular function by non-genetic means through its physical and structural properties, particularly by exerting mechanical forces and by serving as a scaffold for binding of cellular components. The Global Genome Initiative, a collaborative effort to sequence at least one species from each of the 9,500 described invertebrate, vertebrate, and plant families is one of many such ventures. The information from these various efforts is collected in enormous online repositories so that it is freely available to scientists.
Genomics and proteomics field. Genomics. Genomics is the study of whole genomes of organisms. It involves the study of all genes at the DNA, mRNA, and proteome level, as well as the cellular or tissue level. By definition, it can be defined as a discipline in genetics that applies recombinant DNA sequencing methods, and bioinformatics to sequence, assemble, and analyzes the structures and function of genomes. Genomics led to proteomics via transcriptomics as a logical step. Proteomes can be studied using the knowledge of genomes because genes code for mRNA and the mRNA encode proteins. Proteomics complements genomics and is useful when scientists want to test their hypotheses that were based on genes. Even though all cells of multicellular organisms have the same set of genes, the set of proteins produced in different tissues is different and dependent on gene. There are also protein-protein interactions, which complicate the study of proteomes, although the genome provides a blueprint. The final architecture depends on several factors that can change the progression of events that generate the protein.
Proteomics. Proteomics is the large-scale study of proteins. It has enabled the identification of ever-increasing number of proteins. Proteome is a set of proteins produced in an organism, system, or biological context. Proteomics relies on three basic technological cornerstones that include a method to fractionate complex protein or peptide mixtures, MS, mass spectrometry, to acquire data necessary to identify individual proteins, and bioinformatics, to analyze and assemble the MS data. Number 1. Proteomics has multiple methods to study proteins. Generally, proteins may be detected by using their antibodies, immunoassays, or mass spectrometry. Mass spectrometry is a key technique for protein or peptide and PTM analysis. Number 3. Bioinformatics is a subdiscipline of biology and computer science concerned with the acquisition, storage, analysis, and dissemination of biological data. Proteomics has three types. Number 1, protein expression proteomics. Number 2, structural proteomics. And number 3, functional proteomics. Protein expression proteomics is the quantitative study of protein expression between samples that differ by some variable is known as expression proteomics. Number 2, structural proteomics aims to generate useful 3D structures of the entire proteomes using a combination of experimental structure, determination, and modeling. Number 3, functional proteomics aims for the elucidation of the biological function of proteins or protein groups and classes on a proteome-wide level. This includes the characterization of enzyme activities as well as protein interactions and post-translational.
Good morning everyone, I am Kathlen Joy TICMAN and I am going to report about genetic engineering. Genetic engineering. Genetic engineering refers to the direct manipulation of DNA to alter an organism's characteristic or phenotype in a particular way. Sometimes called genetic modification, it is the process of altering the DNA in an organism's genome. It may also mean that extracting DNA from another organism's genome and combining it with the DNA of that individual. Genetic engineering is used by the scientists to enhance or modify the characteristic of an individual organism. Genetic engineering can be applied to any organisms from a virus to a sheep. For example, genetic engineering can be used to produce plants that have a higher nutritional value or can tolerate exposure to herbicides. How does genetic engineering works? To help explain the process of genetic engineering, we have taken the example of insulin, a protein that helps regulate the sugar levels in our blood. Normally, insulin is produced in the pancreas, but in people with type 1 diabetes, there is a problem with insulin production. People with diabetes therefore have to inject insulin to control their blood sugar levels. Genetic engineering has been used to produce type of insulin, very similar to our own, from yeast and bacteria like E. coli. The genetic engineering process. The first one is a small piece of circular DNA called plasmid is extracted from the bacteria or yeast cell. Second, a small section is then cut out of the circular plasmid by restriction enzymes called molecular scissors. Third, the gene for human insulin is inserted into the gap in the plasmid. This plasmid is now genetically modified. Then fourth, the genetically modified plasmid is introduced into a new bacteria or yeast cell. Fifth, this cell then divides rapidly and starts making insulin. 6. To create large amount of the cells.
I am Pauline TABLADA, and I'll be presenting the examples of genetically modified organisms. Let's start with what are the genetically modified organisms. Genetically modified organisms can be defined as organisms such as plants, animals, or microorganisms in which the genetic material or the DNA has been altered in a way that does not occur naturally by mating and or natural recombinant. The technology is often called modern biotechnology or gene technology, sometimes also recombinant DNA technology or the genetic engineering. So, agricultural plants are one of the most frequently cited examples of genetically modified organisms. Some benefits of this genetically engineering in agriculture are increased crop yields, reduced costs for food or drug production, reduced need for pesticides, enhanced nutrient composition and food quality, resistance to pests and diseases, greater food security, and medical benefits to the world's growing population. In addition to that, advances have also been made in developing crops that mature faster and tolerate aluminum, boron, salt, drought, frost, and other environmental stressors, allowing plants to grow in conditions where they may not otherwise flourish. So these are the examples of genetically modified plants. Number 1, soybean. It has a genetically conferred trait of herbicide tolerance. Glyphosate herbicide tolerance conferred by expression of a glyphosate-tolerant form of the plant enzyme. Number 2 is BT corn. It has a genetically conferred trait of insect resistance. It is the resistance to insect pests, especially the European corn borer, through expression of the insecticidal protein. To create a BT corn crop variety, plant scientists select the gene for a particular BT toxin and insert it into the cell of the corn or cotton plant at the embryo stage. The resulting mature plant has the BT gene in all its cells and expresses the insecticidal protein in its leaves. Number 2 is the canola. It has a genetically conferred trait of altered fatty acid composition. It is achieved more effective weed control, reduce overall pesticide use, and improve farming practices such as enhanced conservative tillage, lower list of herbicide resistance developing, and a lower environmental footprint. Number 4 is the plum. It has a genetically conferred trait of virus resistance. It is the resistance to plum pox virus conferred by insertion of a coat protein gene from the virus. To name a few, now that this plant is immune to viruses, there will be a greater production of them. This causes the price of the plums to plummet. Not also thus it reduces the price, but can sustain its quality. They can be kept on shelves longer now, therefore making less food go to waste. Genetically modified organisms can also make this plant capable of surviving in the different environments. So this is an image of genetically modified soybean from Ukraine, a comparison between the BT corn and regular corn, a genetically modified canola oil, and a genetically modified plum. So after the plants, we have also a genetically modified animals. And why are animals being genetically engineered? As scientists have sequenced the genomes of the domestic animals, more is known about genes and the traits that they control. By finding genes that control beneficial traits, we are able to precisely introduce those genes in the another animal's genome, so the GE animal will possess that trait. So an example of genetically modified animal is a genetically engineered aqua advantage salmon alongside an Atlantic salmon of the same age. This fast-growing salmon is genetically modified salmon that grows to market size in half the time as a conventional Atlantic salmon. This GM salmon grows to market weight in about 16 to 18 months versus a 32 to 36 months for conventional salmon. Research on genetically engineered salmon started in the mid-1980s after years of research access to the growth hormone gene and identifying the DNA sequence. The first transgenic was created in the 1989, but it wasn't until almost 20 years later that the FDA approved this genetically altered salmon. So this salmon has been developed to actually benefit the environment. For example, it conserves wild fish population, it reduces carbon emissions, provide low-impact fish farming, and many more. After learning the benefits of the genetically modified organisms, these are the main issues of concern for human health. While theoretical discussion have covered a broad range of aspects, the three main issues debated are the potentials to provoke allergic reactions, allergenicity, gene transfer, and outcrossing. Allergenicity. As a matter of principle, the transfer of genes from commonly allergenic organisms to non-allergenic organisms is discouraged unless it can be demonstrated that the protein product of the transferred gene is not allergenic. While foods developed using the traditional breeding methods are not generally tested for allergenicity, protocols for the testing of the GM foods have been evaluated by the Food and Agriculture Organizations of the United Nations and the WHO, which is the World Health Organization. So far, no allergic effects have been found related to GM foods currently on the market. Number 2 is the gene transfer. Gene transfer from GM foods to cells of the body or the bacteria in the gastrointestinal tract would cause concern if the transferred genetic material adversely affects human health. This would be particularly relevant if antibiotic-resistant genes used as markers when creating GMOs were to be transferred. Although the probability of transfer is low, the use of gene transfer technology that does not involve antibiotic-resistant genes is encouraged. Last is the outcrossing, the migration of genes from the GM plants into the conventional crops or related species in the wild, referred to as outcrossing, as well as the mixing of crops derived from the conventional seeds with GM crops, may have an indirect effect on the food safety and food security. Cases have been reported where GM crops approved for animal feed or industrial use were detected at low levels in the products intended for human consumption. Several countries have adopted strategies to reduce mixing, including a clear separation of the fields within which GM crops and conventional crops are grown. So that would be all. Thank you for listening.